Good morning, dear students. Today we will talk about the beta oxidation. Um, we the fat or lipid from dietary sources gets hydrolyzed to give fatty acid and glycerol. So that fatty acid, you could see here, undergoes oxidation via different steps. So we are having many type of oxidation for the fatty acids, may be saturated or unsaturated, like beta oxidation in mitochondria, alpha oxidation in endoplasmic reticulum or peroxisomes, and omega oxidation in endoplasmic reticulum or microsomes. So all besides other type of oxidations. So you have to study the beta, alpha and omega type of oxidation. So in the present session we are discussing about the beta oxidation. So beta oxidation, this is a spiral event where a fatty acid after its oxidation is converted into a form which is having carbon chain less two smaller carbon chain in number. Okay, so the major steps for the beta oxidation are: the first step activation, then the second step dehydrogenation, third one is hydration, then dehydrogenation, and the last step is cleavage to give acetyl coenzyme A plus fatty acid, which is 2 carbon shorter in nature. So what are the 5 steps? These are the 5 steps that we could see during the beta oxidation, and again activation, dehydrogenation, hydration, dehydrogenation and cleavage to give acetyl coenzyme and fatty acid. Now what are the enzymes that play their role during each and every step? For the first step we are having thiokinase, for the second step we are having dehydrogenase, for the third step enoyl hydrase, and for the last step beta keto reductase. So coming back to the spiral of the beta oxidation, here we could see this is the fatty acid obtained after the hydrolysis of triglyceride, and this fatty acid, the first step is what? Activation. This after the first step, this fatty acid is converted into, you could see, fatty acyl coenzyme A derivative, and during the second step this fatty acyl coenzyme A derivative is converted into alpha and beta unsaturated fatty acyl coenzyme A. This step is what? Dehydrogenation. So you could see the unsaturation at the alpha and beta position, and this alpha and beta unsaturated fatty acyl coenzyme A is, after the third step, is converted into this beta hydroxyl fatty acyl coenzyme A. Beta hydroxy fatty acyl coenzyme A derivative after the step 3. So the third step is hydration, addition of water, you could see here, and after this step, after step 3, we are having the beta hydroxyl fatty acyl coenzyme A. This beta hydroxy fatty acyl coenzyme A, after the step 4, that is dehydrogenation, you could see the removal of hydrogen. Dehydrogenation is converted into this structure. This is known as beta keto acyl coenzyme A. Now this beta keto acyl coenzyme A, after the step of dehydrogenation in the presence of enoyl hydrase, is having beta keto fatty acyl coenzyme A. This shows the cleavage. You could see I have tried to isolate one of the component after this, after the step 5. The step 5 is having the cleavage step in the presence of this beta keto beta ketothiolase. Here, this is the acyl coenzyme A derivative. You could see this structure will be converted into acyl coenzyme A after the presence of your enzyme beta ketothiolase. So this is the first step, and during this we are having the final product acyl coenzyme A and acetyl coenzyme A. You could see here. So this acyl coenzyme A derivative is like the second step of the last chain fatty acyl coenzyme A, but this is having 2 carbon less than the earlier one. This structure is having 2 carbon less than the initial earlier structure. So and this acyl coenzyme A again will undergo many spirals until the entire chain of the fatty acid is converted into the acetyl coenzyme A. Now this acetyl coenzyme A will further will be utilized into the TCA cycle. So during beta oxidation we are having the 5 major steps: activation, the first step; second, the dehydrogenation, converting the fatty acid in the fatty acid into fatty acyl coenzyme A, and then fatty acyl coenzyme A into alpha beta unsaturated fatty acyl coenzyme A after the step second, and this alpha beta unsaturated fatty acyl coenzyme A after the hydration event of hydration is converted into beta hydroxyl fatty acyl coenzyme A, and this beta hydroxy fatty acyl coenzyme A after the step 4, that is dehydrogenation, is converted into beta keto fatty acyl coenzyme A, this is structure, and this beta keto fatty acyl coenzyme A is undergoes the event of cleavage in the presence of beta keto thiolase to be converted into the acyl coenzyme A and acetyl coenzyme A derivative. So we could see that during the first step, step number 1, what happens? The fatty acid is activated and converted into thioester by thiokinase in presence of our coenzyme A, ATP and magnesium ion. Then the above fatty acyl coenzyme A, the thioester is dehydrogenated. You could see here, dehydrogenated, leading the unsaturation between alpha and beta. Alpha and beta carbon atoms. This happens in presence of enzyme dehydrogenase and FAD. This unsaturation happens in the presence of FAD and in the presence of enzyme dehydrogenase. So after the step 3, we are having the event of the hydration, and this hydration, in the next step, the alpha beta unsaturated fatty acyl coenzyme A is hydrated in presence of enzyme enoyl hydrase via attaching hydroxyl to beta carbon and a hydrogen at the alpha carbon. You could see here. Then in the next step, the beta hydroxyl fatty acyl coenzyme A is dehydrogenated to give beta keto fatty acyl coenzyme A in presence of enzyme dehydrogenase or beta keto reductase and DPN plus. In the last step, step 5, what happens? This beta keto fatty acyl coenzyme A undergoes cleavage step, and in this step, there occurs a cleavage between the alpha and beta carbons, that is thiolysis involving coenzyme A. You could see here coenzyme A and enzyme thiolase or beta ketothiolase to give acetyl coenzyme A. You could see here acetyl coenzyme A and fatty acyl coenzyme A. Fatty acyl coenzyme A with two less carbons. This two carbon is smaller. Fatty acyl coenzyme A again enters the same cyclic process. You could see the same cyclic process 1, 2, 3, 4 and 5 as a new spiral. The event continues. This event means this event of spiral continues up to the conversion of fatty acid completely into acetyl units. If there is suppose 18 carbon fatty acid in the beginning, in the before the first step, if you are having 18 carbon fatty acid, that will ultimately give 9 acetyl coenzyme A. So this is whole about the beta oxidation. For the in case of fatty acids with odd number of carbon atoms, removal of acetyl units leaves at the end a propionyl coenzyme A residue, and this may be converted into propionic acid in heart and kidney tissues, and in the liver the propionic acid does not accumulate but is oxidized to carbon dioxide and water, involving formation of succinyl coenzyme A and Krebs cycle. So this is whole about the beta oxidation. So we are having the 5 different major steps: activation, dehydrogenation, hydration, dehydrogenation and cleavage, and the enzymes used for these different steps: thiokinase, dehydrogenase, enoyl hydrase, beta keto reductase, and lastly beta ketothiolase. So the fatty acid is first converted into fatty acyl coenzyme A, and this fatty acyl coenzyme A is converted into alpha beta unsaturated fatty acyl coenzyme A derivative, and this alpha beta unsaturated fatty acyl coenzyme A after the step 3 is converted into beta hydroxyl fatty acyl coenzyme A, and this beta hydroxy fatty acyl coenzyme A, we could see in the figure, is converted into beta keto fatty acyl coenzyme A derivative. And after this step, after the step 4, we are having the event of cleavage, where this entire structure is converted into a fatty acyl coenzyme A derivative plus acetyl coenzyme A, and this fatty acyl coenzyme A is having having a chain which is having two carbon shorter chain. It means in this acyl coenzyme derivative there will be two less carbons than the first one after step 1, and this fatty acyl coenzyme A derivative again will enter in a new spiral, and this different turns will be continued up to the conversion of entire fatty acid in the beginning into the acetyl coenzyme derivative, and after each spiral the new acyl coenzyme derivative will have the a chain which is having two carbon less. So this is whole about the beta oxidation, and this is definitely in or might be in your question paper, that what is beta oxidation, discuss its different steps in detail, and for this you have to always keep it in mind that these are the major steps and these are the enzymes, and during each step there will be the role of this particular enzyme. So add the role of the enzyme during each step and the other elements present and the conditions present in order to convert that initial compound into the next step. And keep it in mind that always this spiral will result into a acyl coenzyme derivative having two less carbon atoms and a acetyl coenzyme A, and this step will be continued up to the conversion of initial fatty acid into the acetyl coenzyme A. So that's all about the beta oxidation. In the next session, we will discuss about the omega and alpha oxidation. Thank you.